So we're kicking off the next session. And so this is an aggregation across chemistry, instrumentation, and agriculture, so session 4. And Agilent Technologies, in particular Craig Marvin from Agilent Technologies will kick it off with a keynote. Craig received his bachelor's in animal bioscience from Penn State University and then performed graduate studies at Purdue, and then joined Agilent. Prior to his current role at Agilent, he served as Agilent's Global Environmental and Industry Manager, responsible for business development and strategic marketing activities in the environmental industry. So I think as part of that extensive background, he'll be able to share with us today his thoughts. He spent his 20-plus years of career focused on environmental analysis applications, served on a number of advisory boards, and has been leading a lot of the strategy within Agilent. So Craig, thank you for representing Agilent, and we look forward to your words and remarks, and we'll turn it over to you.
All right, Brian, thanks very much. And thank you to everybody. I know it's right after lunch, so I'll try to be a little bit more dynamic so we don't lose anyone for sleeping. I would like to thank the organizers for allowing Agilent to come and present. I'm actually here as a surrogate for Darlene Solomon, who is a PhD alumni from MIT, and she apologizes that she couldn't be here. She had a previous commitment, so she asked me if I'd come and speak to you. What I'd like to talk about is the challenges of trace chemical monitoring. Part of what I was asked to do is to talk about technology, and I really didn't want to get into an instrumentation presentation, but I'll talk about applications in areas where I think this community could probably put its head together and help the industry as well as the regulatory community come up with better options for trying to monitor chemical contaminants in the environmental space. So the first thing I want to do is to wish everyone a happy Environment Day. How many people in the audience knew that the UN has June 5th as the Happy Environment Day? Okay, well, congratulations. Essentially what they're asking, very few hands went up, but that's okay. Essentially what they're trying to get the global community to do is to organize and become united, and in 2018, let's beat plastic pollution. We heard a presentation earlier about the microfibers and things like that. But in the ocean, what you find is there are a lot of microplastic materials that have a negative impact not only on water quality, but also on biota. Fish, animals eat this material, disrupts their digestion, et cetera. And we heard a comment in the first presentation about what's happening in the reefs around, the Great Barrier Reef, et cetera. This has a huge impact on the Great Barrier Reef as well as the nanoparticles that we'll talk about here in a little bit. So happy World Environment Day. I found it rather fortuitous that we're having this conference today, but we'll move forward and see what we can do. So there are a number of environmental issues that are of global importance. The population is growing, projected to be about 10 billion people by the year 2050. If we look at the largest populated country on the face of the planet, they're already facing a challenge because 25% of their arable land is unfit for utilization. As they continue to change over from industrial utilizations to try to go to arable or inhabitable land, they face a huge challenge. There's a huge initiative there to try to understand what is the source of this contamination and how can they address it. The other thing that you need to consider is that there's more than almost 700 million people around the globe that don't have access to clean water, or at least access to clean water that's close to them. But on top of that, with the growing population, the UN projects that by 2050, the demand for water is going to increase by 55%, okay? By the end of 2030, we will already face a 40% shortfall. So water is probably the most precious resource that we have on the planet. And you'll notice here, and I'll talk about this a little bit later, but I pulled this last night from the web. So this is the Chem Abstracts CAS registrations. 142 million chemicals have been registered with CA Select. Okay? If we look at what goes on, agriculture, the environment, food requirements, what you'll notice is that it's a very difficult scenario. We need to have agriculture to sustain life on the planet, but the things that we do to maximize yields and make sure that we're producing beautiful fruit, beautiful vegetables that people like to eat cause problems. So to support this 7.6 million population, we have a very significant impact on the environment, processing, runoff of herbicides and pesticides. And of those 142 million chemicals, 65 are currently commercially available. Now, those of you who may be familiar with EPA methodologies, MEP methodologies, or things that are put forth in Europe under CEN, should realize that we monitor a fraction of a percent of those 65 million compounds. So those individuals who are very concerned about chemical contamination and its impact on human health or biological health in the environment face a really difficult struggle. And we'll talk a little bit later about strategies that can be employed to try to facilitate that process. Instead of trying to look at everything, how can we determine the chemicals that we really need to be concerned about because they're going to have some biological or negative activity on the biota? So I work for Agilent Technologies. We're engaged in a variety of segments: pharmaceutical, biopharmaceutical, clinical diagnostics, food, energy and chemical, forensics, toxicology. But what I'll talk to you today is the environmental segment. We have a long history, 50 years of working in this segment, and for about 40 of those years, we've been the market leader in water analysis. Okay, so our team, working with around the globe with global partners, key opinion leaders, and collaborators, continue to drive research innovation, technology innovation in this particular area of research. The goal of what we're trying to do is to come up with better techniques, better opportunities, better applications to use trace chemical monitoring in the environment that allow us to protect human health as well as the biological health. So a lot of this work, as I said, is done through collaboration. And I know that Darlene, with her commitment to MIT, is working to develop or foster those type of relationships here at MIT. But what you see on the screen before you is sort of a global map of the current status of collaborations around the globe. You'll notice that there are a variety of companies: Dow Chemical, Metrome, an instrument company that we partner with to hyphenate techniques to provide strong analytical capabilities to the research and monitoring community. We work with a number of universities around the globe, have collaboration with the China MEP, their analytical method development, to help ensure that in that expanding and growing economy, they're able to have access to technology and applications that help them to develop methods suitable for the challenges that country faces. So historically, we've been known to support this analytical chain of events within the environmental segment. And most of this involved quantitative analysis. We made our reputation on GC mass spec, which is used for organics analysis, volatiles, semi-volatiles, pesticides, herbicides. And that has slowly migrated into LC techniques, ICP-MS. But where we've seen things moving is into this identification of compounds that people weren't expecting to see. Okay, quantitative analysis involves a target list. If you look at EPA method 8270, 137 compounds, 524.3 for volatiles in drinking water, about 130 compounds. The big concern is when you're looking at these environmental samples, what is it that you're not missing? And so what we see in the industry is a transition to high-res accurate mass spec techniques that allow you to essentially ionize everything that's in that sample and then use selective detector technology as well as software tools to be able to extract the ions, remove matrix interferences, and identify compounds that should be of interest. Couple this with this omics-based research, proteomics, metabolomics, et cetera, or exposure biology, where we actually expose biota, could be fish eggs, could be cells, could be fish larvae, to a chemical challenge in a surface water, in a wastewater, and we analyze those samples that demonstrate some biological activity, where we see either a reduction in metabolic processes within the biota, or we actually see some mortality. That indicates to us that that water sample has a chemical that's present that may have a detrimental effect to the human population. And then we can deploy these high-res, accurate mass technologies. So Agilent is a company, we're very well known for technology innovation. And with this new high-res accurate mass, we've been challenged by the research community to say, can you give us a better tool? Okay, when we look at this cischlordane, okay, With the earlier technologies, this is kind of what that ion profile would look like. It looks kind of like a humpagram. You know, there are things there, but it's very difficult to distinguish what's going on. So there were some real distinctive innovations made to the detection cell, where now with the new 7250, what you're able to see is that we're able to break that humpagram into 4 distinct ionic components. The thing that's interesting about this is that we have very good mass resolution. These compounds don't differ very much through molecular weight or through their ion profile. And essentially what you find is those 4 compounds that are on the right are actually the components of that humpagram. This actually changed the game for these people involved with non-targeted screening and unknowns analysis, because it gave them a very powerful tool for trying to characterize what's going on in these sample matrices. This particular technology has been applied for dioxins type analysis. We're hoping that it should be able to replace high-res accurate mass spec. This is a technology very expensive, very difficult to maintain, and the EPA and the MEP are looking at alternates to this to allow you, the researchers, to 1, be able to screen more efficiently, and then 2, to confirm when you have something that shows up. So here we have a compound that there was an abundant brominated unknown that was found in some sediment based upon this ion profile that we see up on the right. So we go out and look at in ChemSpider and come up with a 1,3,6-A tetrabromomethyl carbazole compound. And as we go through the iterations of software analysis, et cetera, what we come up with is that we actually are able to confirm that this is the structure. Once a researcher would say, hey, we think we have this structure, what they would do is go obtain that compound and simply do the analysis again to make sure that this ion profile matches. If it's a match, now they can start developing quantitative methods to help the regulators understand the distribution and the profile of this particular component in the environment. So I'm going to talk about some application areas, some things for you to think about where some of this nanotechnology and some of the sensor technology that was discussed this morning may have application for real-world requirements. The first is this direct potable reuse. Those of you who are familiar with what's going on in the United States and other parts of the world, the southwest portion of the US has been under severe drought conditions, as well as Texas and Florida. And if we look at what happened in 2013, Governor Brown put a dictate in front of the state water board saying that California will have direct potable reuse by the year 2016. Our collaborator, Shane Snyder, Agilent thought leader at the University of Arizona, spoke about the stresses of freshwater resources and the need to look for alternative sources of water supply. The first speaker today spoke about how we need 4 globes to be able to support the future growth of the population. It's going to come quicker than that. I think we're going to have to really look at how we can improve or sustain, or I guess you could say save water, the most precious component on the face of the earth. It's not only in the United States. Singapore has been using recycled water for many years. If you go to the Middle East, they do some desal. They're also looking at recycled water. It has a little bit of a bad connotation. But the one thing I always try to get people to understand is currently most of the water that's consumed by the human population undergoes what's called indirect potable reuse. So my joke to all my colleagues when I talk about DPR is you're always downstream from somebody, okay? So what happens, if we look on the top here, so someone in Santa Ana, California flushes their toilet, it goes into their sewage treatment facility in Fountain Valley, and it produces a liquid that's cleaner than rainwater, okay? That water then gets pumped in 13 miles away from Santa Ana and goes into an aquifer where it, quote unquote, goes under biological processes to further clean and clarify the water. Well, what ends up happening? It actually gets contaminated. It's dirtier when it comes out of that aquifer than it was before it went in. And then from that aquifer, the local community will pull water and purify it for drinking water to feed 2.3 million people, okay? Under direct potable reuse, that whole program changes. Essentially, we don't send the water out for environmental conditioning. Instead, we directly connect the outlet flange from the wastewater treatment plant to the inlet flange on the drinking water plant. Now, you're not going to drink solely reused water. It's used to supplement the water that's pulled from the ground or the aquifers. And it allows us to sort of keep water in the cycle and retain it as opposed to discharging it into the rivers and ultimately into the ocean, et cetera. This reuse also takes place with respect to agriculture, and we'll talk about that here in a little bit. The concern here is that wastewater treatment isn't 100% effective in removing all the chemical contaminants out of the water stream. Here on the lower right, you see the composition of an average wastewater treatment plant in the United States. A lot of artificial sweetener, pharmaceuticals, stimulants, X-ray media, et cetera. And these other three, this was work that was done by a colleague of mine, Tarun Animal, in Chennai, looking at the 3 wastewater treatment plants in India. And you'll notice they also suffer from having X-ray contrast media, stimulants, and pharmaceuticals that show up in the water that gets released out into the natural waterways that then are used to provide source water. So the big concern is with direct potable reuse, if we have this going on through that indirect, what's going to happen when we're in the direct potable reuse? So we underwent a project with a small university in northern PA, and the goal of this project was really to look at fracking intrusion on natural waterways. But what we found out, just because of the natural gas price, they stopped production. So they held off. So we salvaged this by trying to go out and look at what happens. Can we look at runoff from the rural waste disposal systems as well as a waste treatment facility, the septics, et cetera. So we had this team went out and they collected over the course of a year, over 8 months. And essentially the workflow was that we would get these samples, we would go through a separation, LC separation with an online solid phase extraction for a little bit of enrichment. We would then take the eluent into a QTOF to ionize all the species that were there. We would use a software, ProFinder molecular feature extractor, to identify compounds or structures that were in that sample that we could recognize. And then we would compare that to a compound database library where we've collected 2,000, now it's up to about 5,000 spectra. But what that allowed us to do is to go in and selectively say, hey, we found these compounds. That's the easy part of the job. We then would go in and use a molecular structure correlator to go in and say, okay, we found hypothetically 96 out of 100 compounds we could identify. What are those other 4? And that's where the unknown analysis comes in, and that's the challenge of using these high-res accurate mass techniques. So along the top here is kind of what you see. So we started with the wastewater effluent, and you'll notice we extracted 3,000, the group in Mansfield extracted 3,000 features. Going through the QC and the subs and everything, we were able to identify 746 features. And doing a PCA, they were grouped with blanks and controls, things that were upstream from the wastewater, things that were in the wastewater effluent, and things that were downstream from that wastewater plant. So we're able to characterize where these compounds come from. When we look at an 8-month moving average, we were actually able to find that there were 141 compounds that were sort of unique, features that were unique to what was going on in that aqua system around Mansfield, Pennsylvania. So as we went down and continued, we looked at the wastewater effluent features. We found 31 unique features. And then we had 5. We were out trying to find unknowns. 5, we found a variety of carbamazepine, valsartan, et cetera. And then there were a few that we ended up having to do with this structural correlator to find, this benzothiol, et cetera. And what this demonstrates is the power of this tool. The challenge, though, is that this is a very laborious process. It took 8 months. So if you think about direct potable reuse, where we're connecting the outlet flange of that wastewater treatment plant to the inlet flange of the drinking water treatment plant, we need something that's online and real time in order to characterize what's taking place. So being able to develop these sensors that everyone is talking about this morning is a powerful tool for this community. And this is kind of what this would look like. So this is the San Jose DPR plant. So this was the first drinking water, direct potable reuse plant that was in operation in California. It's down the road from our headquarters in Cupertino. But what you see here are some sensor arrays, and these are nothing more than nitrate, nitrite, turbidity, conductivity, things like that. No organics are being monitored. There may be a total organic TOC number that comes out of here, but there's nothing specific to tell you what's composing this water. So the assertion that we see in the industry is that this initiative, and it will continue to grow exponentially around the globe, will drive the demand for at-line or online type monitoring systems. So this is a challenge for the research community. I mentioned before the use of chemical exposure, looking at biological response for emerging contaminants. And what this allows you to do is you've got a chemical origin, microbial origin, and then there's a workflow that we've spoken about, sample prep. But the one thing that we insert here is some type of a device that allows us to characterize biological response. This is a Seahorse cell metabolism, and it's manufactured by a company in Lexington just down the road here. And what this does is allows you to expose cells and monitor their metabolic activity. When you see changes in that metabolic activity, you've identified that sample that I was talking about that has an impact on the ability of that organism to survive. And so usually this is built into three components. The Seahorse to look at the metabolism of the living cell. Once they're done, you can use the cells for something else, much like what we heard just before lunch. We might do genomics. We would use a bioassay. The EPA and NHS or NIEHS are working to come up with gene arrays that are indicators that we can use to challenge against a matrix and look for a heat map and see where we see activity. And then the metabolic profiling allows us to look at the biochemical phenotype of what's going on in that particular sample. Okay? Our collaborator I mentioned before, the Snyder Group out of the University of Arizona, has done some very interesting work. Very often when we hear about environmental contamination, everyone thinks it's bad. Someone's going to get sick, someone's going to get ill, someone's going to get cancer. They identified something that was happening, a unique thing going on in the Amish communities in central Pennsylvania, where they noticed that there was a lower incidence of asthma among teenagers, preteens and teenagers in that community than there was in the global community for teenagers. So they went out to investigate what's going on that protects the lungs of these individuals from asthma. So they went through a series of things. They looked at a variety of different things. They were, looking at hay dust and extracts and things like this. And so they had some mice that they exposed to a control. They thought it was biological, house dust mites, et cetera. And then they positioned house dust mites with this Amish extract, what we see up here. And they noticed that there's something in that structure of the Amish extract that has an impact on the epithelial cells. So they went forward through a variety of techniques, size exclusion chromatography. And essentially what you see here is the chromatography and then a heat map of activity using one of these bioassays, challenging the epithelial cells of the lungs in these mice. And in this fraction, they went out and then tried to say, what can we do using these high-res accurate mass and a variety of techniques to identify what's that chemical structure? What gives us that protective environment? And so they worked with cell, went through a variety of things, but they went through essentially a full profile. They used everything that was in their arsenal, the QTOFs, LC QTOF, they went through peptide mapping, they went through peptide ID through MSMS, they did some sequencing to identify what was going on. And what's interesting to me is that this was funded by a major pharmaceutical company. And when this group started, the pharma company said, you will never succeed because you only have a lowly QTOF. We have, a competitive product to Agilent, which I'm almost forbidden to mention. But they were very surprised because they were getting ready to pull the plug on the project until Shane showed some data that gave them some indication that in one of these fractions, this dB or DC fraction, this is grouped enough where they've been able to identify there's one of 3 compounds that are in these fractions that are causing this impact. And the company said, we need 5 minutes. Everyone leaves the room, and they came back and refunded the research project. So these type of things, I think, are tools also where being able to understand and identify through nanotype technologies what's in the environment might have a beneficial effect. We spoke about agriculture, okay? The first speaker spoke about spray and pray type applications of herbicides and pesticides in the agricultural community. There is an initiative around the globe to utilize engineered nanomaterials to do some strategic positioning of fertilizers and things at the root level. We're doing work with Arturo Keller here in the middle in blue. He's also an Agilent thought leader, and he manages the University of California Environmental Implications of Nanotechnology group. And essentially, he has been working to understand the ability a lot of work in his previous research, understanding the impact of nanoparticles in the environment, and now working on trying to look at the strategic placement of nanomaterials at the root structure, understanding their fate in the environment, as well as do they show up in the fruit and vegetables of the plants in which these are being used. And one of the tools that's really helped them is this single nanoparticle analysis by ICP-MS, where you can get a raw signal and then characterize not only the size, but also the type of nanoparticle that's included in that particular structure. Arturo provided me with this slide, and it's quite interesting to understand nanoparticles are everywhere. We heard that this morning. You know, we have silica oxide that's used in automotive. We have zinc and titanium oxide that are used in personal care products, sunscreens, et cetera. Copper oxide. Interestingly enough, copper oxide is considered an organic pesticide for food production because it occurs naturally. Okay, so people apply this, but there's really no understanding as to what the impact is on the human body. There was a mention about, you know, the impact on the Great Barrier Reef. If you try to go diving there now, they're going to ask you not to use sunscreens that you bought commercially from the shop at your hotel before you went out on the boat. It's this Ti oxide, the zinc oxide, and all of these have a little bit of silver oxide, which has a detrimental effect on the flora of the Great Barrier Reef. So they're really trying to protect that. So there's a lot of work done with silver nanoparticles. These are used as antibacterials, et cetera. You see them a lot on gym clothing and everything to prevent that funky smell if you don't empty your gym bag. And this is just looking at the appearance of these silver nanoparticles in influent water, effluent water, backwater, and filtered water. And what you'll see there is a degradation, but it's this appearance of this silver plus ion that causes the detrimental effects in the biological substructure of the water system. I mentioned the use of this copper oxide. So there was some work done where there is looking at 3 different leafy vegetables: lettuce, very smooth leaf; collard green, a little bit rougher; and then kale, which has a very rough architecture. All of these plants have, you know, vesicles or orifices by which they respire with the environment. So this is a portal by which they can uptake nanocarbon or these nanomaterials that are applied to the surface. What you see here is that regardless of how much you rinse these things, and this was an aggressive rinse with a buffer solution over a period of time, you'll notice that within each of these leaves, you still retain a high concentration of this copper oxide nanomaterial. Like I said before, it's not really characterized what the impact that will have on the human population. Another challenge, I think that this group is very interested. I saw a lot of synthesis of, you know, membranes and things like this. Keller's group is also trying to look at green chemistry for the creation of nanoparticles. So they're looking at the ability to use the reducing agents that are in plant tissues to sort of aggregate metallic ions that are in the plant extracts here to be able to create nanomaterials and then work to extract those nanomaterials and you know, stabilize those and essentially characterize you know, the appearance or the development of those using the ICP-MS. So those of you in the audience who you know, work in this particular arena, I challenge you to consider you know, a little bit of greener chemistry. This is a push around the globe between the regulating agencies to reduce the amount of organics that are used in the environment. The last thing I'll mention is sensor technology. I think there's you know, we had the presentation just before lunch, which I thought was fantastic. But those of you who are from China or have been to China probably have seen this type of a profile somewhere in a building. There are a lot of sensors that are put around Beijing, Shanghai, Guangzhou, et cetera, where they look at PM 2.5 and a variety of other indicators. So on a good day, you'll notice you get this what they call the APEC blue sky. The sky is blue, there's no haze. This was actually 2 days later from this picture, and you'll notice that everything has deteriorated. The composition of this PM2.5, which causes that haze, has a very complex organic component composition. So what the China government is looking for are sensors that would allow them to understand or identify the sources of these organics that coat these silica beads that float in the air. Another challenge I'll put forth to you to try to look at things. And it is an expanding technology. The EPA, the MEP, the European Commission are all looking at ways that they can miniaturize devices that they can put in the field to monitor air quality. One of our competitors, Perkin-Elmer, based in Shelton, Connecticut, has the ELM system. The EPA is working on a green bench system. And there's a huge initiative in China to look at fugitive emissions, field fence line emissions, et cetera, using a variety of technologies. So I thank you for your attention. I'm hoping that maybe some of the information I presented today will give you food for thought, some things you may want to think about as you evolve in your research. And if there are any questions, I'll be more than happy to take those at this time. So, Brian, thank you.
Great, thank you. So yes, please, we'll take some questions.
Be gentle. Yes?
Oh, you're not loud enough.
So I'm wondering, when you detect these different organics, and there's going to have to be probably a suite of different technologies to remove them.
Correct.
Right? There'll be, some, like the case you had that was brominated wouldn't burn very well. Right?
Correct.
And so is there a lot of coordination between you and the remediation part, or how best to come up
So
with
what usually happens, so when we look at this, when we identify that there's a problem, okay, we'll work with the regulators, and we'll go through some type of a risk assessment. That's the first step that's taking place. What's the risk to society? If it's a high risk, it gets a priority. And so the priority is they go through the metabolic testing, what's the maximum recommended limit for exposure and those type of things. And once that's done, then they start working with researchers. So either industrial researchers, it could be 3M, it could be Calgon or these other companies, or university researchers to understand what technologies would be available to help remove this. There's a lot of work that's being done with nanoparticles to see, can we encapsulate nanomaterials with chelators that would remove metals, et cetera, and where we can reverse those. Or can we actually have something that binds to these organics that have, say, a metallic core, and that as we go through a water treatment, we can simply turn a magnet on and pull all the nanoparticles to the side, send clean water out, pump the nanoparticles out to regenerate those and go through that process. So it is a long, iterative process. The EPA makes a glacier look like Usain Bolt, so they move very, very slow. And it's a long, laborious process to get from detection to what are we going to do to remediate it.
Other questions? So I had a question just because I've spent a lot of time in Singapore. So why are some, other than some resource constraints, a country like Singapore is far ahead of where we are in sort of direct?
Yeah, so they do not have a large supply of fresh water in Singapore. And even if you go back to World War II, one of the reasons that they capitulated during the war to just be sort of neutral, along the causeway that connects Singapore to Malaysia, there's been these large tubes, ducts that carry fresh water to Singapore. So because of that, it's very expensive. Water's very expensive. So they are just trying to be, green citizens and conserve. Unlike the United States, where we had a lot of lakes and a lot of groundwater, Singapore doesn't have that advantage. They're sort of locked by seawater, and anything that's below the ground has a higher incidence of saline content than what you would expect to see, for example, in Indiana or Illinois or something like that.
So how are they a lead user of technology that is sensing that cycle? Or,
yes.
So
yeah. So there are major universities, NTU and NUS are both involved with, and they're really trying to also characterize, what is the chemical composition and what can we do to remove those chemicals? Do we need to do reverse osmosis, membrane filtration, or other technologies to try to ensure that that water remains clean? So there's a lot of investment being made and a lot of work being done. And the government spends heavily to support that activity.
So hand that over there. Here, you can go first.
Hi, thanks for your talk. Is there a way that Agilent is interested in going down to the nano size for detection and purification and so on?
So I will say yes. We realize that there's a real drive within the regulatory and the monitoring community for in situ type measurements. Okay, and in order to do that, you have to miniaturize things. It's virtually impossible to think that you could put a GCIO-TOF or even a GC single quad mass spec into an area where you'd be able to make that work in an outbound mode. So the idea is, what is it that we can do? And I initiated this initiative in my old job. What is it that we can do to work with researchers or to identify the possibility to miniaturize things, put them out for real-time monitoring of a river. Some of this happens in the Danube, in Europe, where they look at changes along monitoring stations, and then report that telemetrically to a central control center. What you're seeing in, for example, with ozone precursor monitoring around the globe, and we didn't talk about air, but in Texas, they actually do this. But unfortunately, right now, they put GCs into a trailer and sample, 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 and then they will send data to a control center so they can look at plumes that are coming from hydrocarbon processing plants, in Texas, California, Oklahoma, et cetera. So yes, there is interest to try to do that. And I think the organization, Darlene Solomon, would be someone who would be very interested to have conversations with researchers who are engaged in that area.
I don't think it was mentioned. I know you mentioned Darlene's name, but Darlene is the CTO of Agilent.
Exactly, Chief Technical Officer, yeah.
Yeah, so my question is actually somewhat related to the previous one, but I see a lot of trends in making smaller instruments, maybe not necessarily at the nanoscale, but I don't necessarily see the big companies like Agilent playing in that field as much as these small companies. So that tells me that there's not a business opportunity there, and yet there seems to be a need from an environmental monitoring standpoint, as your previous answer alluded to. So I'm just sort of trying to reconcile that.
So I think what happens, and I'm speaking as Craig, I'm not speaking on behalf of Agilent. I think when you look at the large companies, we're all driven by Wall Street, all right? What have you done for me lately? What's your quarterly return? How that's going to go? So when you look at these things, there are some challenges when you miniaturize a technique, because some of the detection systems that are currently available require sample enrichment. Okay, if you look at EPA methodologies today, you've got to collect 2 liters of water, you extract it with 200 mils of methylene chloride, and you blow that down into 2 mils that you put into an autosampler vial that you can then monitor, go in and analyze the semi-volatiles. You've just gotten rid of 198 mils of methylene chloride, so you've just created a huge potential for pollution. So what happens is, can we develop microtechnology that allows us to get to those detection limits, where we have confidence that we're going to be able to see or detect at the level that is recommended for a reporting limit in a particular assay. And so I think what you see is that the companies we support through these collaborative researchers, we support people who are investigating that. Helping them, giving them technology and working with them to try to identify what's possible. And I think it's only after something comes out and then goes through the regulatory process of going through its interlab study or validation requirements that the EPA says, oh yeah, this will work, that then the commercial entities will step in and really help startups or innovative research organizations to commercialize these new technologies. Okay.
Okay, I think we probably have time for one more question. So what do you see? So you spend a lot more time talking about water than air, but where do you see some of the challenges for the air and opportunity compared to the measurement of volatiles, organics?
Yeah, so if I were to prioritize, I'd say water is still the primary thing. But there is a drive around the globe to sort of get better control in real time about contaminants in air. The EPA released Method 325, which is a fence line monitoring application. There's a huge initiative in the United States for all 50 states to be compliant with this ozone precursor initiative, looking at things that can create ozone and have some detriment to the protective layer in the atmosphere. And I think what you'll see is that that's probably the second most, at least in this country, the second most important area for future research and development. Of course, in China, I would say that 5 years ago that was the #1 priority because they had a higher incidence of cancer and illnesses related to the inhalation of these PM 2.5 and PM 10 type particles. And they're now undergoing the same type of initiatives as the United States, looking at these volatiles for ozone precursors and trying to understand how they can reduce those emissions, et cetera.
Craig, again, thank you.
All right, thanks very much.